Okay, let's dive right into this explainer and explore the absolute foundation of literally all living things, the cell. I mean, every single living organism on this planet, from the absolute smallest invisible bacteria to a massive elephant, is fundamentally built from these incredible microscopic units.
So, here's our agenda for today. We're going to cover life's ultimate building blocks, discovering the microscopic world, cell diversity and sizes, the plasma membrane, transport, diffusion, and osmosis, and finally, the cell wall.
Section 1, Life's Ultimate Building Blocks. You know, when we look at complex biology, it really helps to frame it just like simple architecture. Just like a single brick builds a wall and the bunch of walls build a room, microscopic cells group together into tissues. Then, different tissues combine to form organs, like, say, your lungs or your stomach. And when these organs work together, they form an entire organ system, like the respiratory or digestive system. Eventually, all those systems come together to build the whole shebang, an entire, functioning living organism.
Section 2, Discovering the Microscopic World.
So, you might be wondering, how did scientists even find these invisible biological bricks in the first place? Well, it all started back in 1665 with Robert Hooke. He put a piece of dead tree cork under a microscope that he actually designed himself, saw these tiny honeycomb-like compartments, and coined the term cell. But it wasn't until a few years later in 1674 that Antony von Leeuwenhoek, using an even better microscope, spotted the very first living, moving cells. And jumping way forward to 1972, we get the fluid mosaic model, which, well, we're going to unpack that one in just a moment.
But really, why did it take humanity so long to find them? Imagine holding a piece of paper with two tiny dots close to your face. As you bring that paper closer and closer, they eventually blur into a single dot. You're actually experiencing your eye's limit of resolution, which is exactly why we need microscopes. For the human eye, that limit is about zero point 0.1 millimeters at a distance of 25 centimeters. If things are closer together than that, our eyes literally just can't resolve them as separate objects.
Section 3, Cell Diversity and Sizes. Now, as you'll see, there is just an incredible variety of cellular shapes and sizes across different living things. But we basically have two main categories of life here. A single-celled amoeba or bacteria is a unicellular organism. It does everything entirely on its own, breathing, eating, moving, you name it. On the flip side, multicellular bodies like plants and humans survive using a massive, highly specialized division of cellular labor.
To put that division of labor into perspective, a single human body contains over 3 trillion cells. 3 trillion! And they are all working in perfect harmony. You've got specific cells to carry oxygen, cells to fight viruses, and cells to send signals right from your brain.
But they aren't all uniform. Actually, cell sizes span some really incredible extremes. While an ostrich egg is the largest living cell on Earth, a tiny bacteria without a cell wall called mycoplasma is the absolute smallest. And inside humans? Well, while the ovum is large and the sperm cell is tiny, human nerve cells or neurons can stretch to an unbelievable 1 meter long. That is a single cell stretched out for a whole meter. Crazy, right? Section 4, the plasma membrane. Okay, let's zoom in on the cell's extremely thin 7 to 10 nanometer outer security gate, the plasma membrane. We really need to dive into this concept of selective permeability. Basically, it means the membrane strictly acts as a gatekeeper. It meticulously controls what enters and exits the cell. It is definitely not a free-for-all. It's a highly secure border.
So how does it actually pull this off? Well, this membrane is beautifully constructed as a fluid mosaic of a phospholipid bilayer. It's made of 2 layers, where the hydrophilic or water-loving heads face outwards toward the fluids, and the hydrophobic or water-fearing tails safely hide inside the layer. Embedded all throughout this are proteins that act as these highly specialized gates and channels.
Section 5, Transport, Diffusion, and Osmosis. So how do substances actually get past this microscopic border control we just examined? The cell uses 2 primary methods. Passive transport, like the diffusion of gases such as oxygen and carbon dioxide, flows freely down the concentration gradient, from high to low. It's easy, like rolling down a hill. But active transport, that forces large molecules against the natural flow, from low to high, and that essentially costs the cell its precious energy. Here's a quick thought experiment for you based on those transport rules. What do you think would fundamentally happen to a cell, or let's say a plump grape, if you placed it in some really concentrated salty water overnight? The answer lies in osmosis, which is a special, absolutely vital type of passive transport specifically dedicated to moving water. Water simply moves from where there's a lot of it to where there's less of it, passing right across that selectively permeable membrane.
Now, these are the rules of tonicity. A fun trick to remember is that hypertonic solutions, which have lots of solutes like salt, make water leave the cell, causing it to shrink. Meanwhile, hypotonic solutions make water rush in, making the cells swell up round. And if it's isotonic, well, the water moves equally, and the cell stays the exact same.
Section 6. The cell wall. Finally, let's look at a cool architectural structure that provides an extra, rigid layer of armor for certain living things. Unlike flexible animal cells, like ours, that only have a plasma membrane, plants, bacteria, and fungi boast a hard outer cell wall that provides crucial structural support against the elements. For their armor, plants build theirs out of cellulose, bacteria use peptidoglycan, and fungi use chitin.
Now, because the outer wall is so incredibly stiff in plants, soaking a plant cell in a hypertonic solution causes the water to rush out, making the inside shrink right away from the wall. It's a visually striking phenomenon called plasmolysis. The wall stands strong, but the living cell inside totally shrivels up.
You know, our lack of cell walls allows for our dynamic flexibility, enabling us to walk, run, and dance. Which leaves us with this provocative question to ponder about the microscopic rules governing our macroscopic lives. If our human cells had rigid cell walls like plants, how would our bodies and daily movements fundamentally change? I mean, think about how completely different your reality would be if you were built like a tree. Thank you so much for joining me on this explainer. Keep wondering, keep learning, and I'll catch you on the next one.